wanted to do just a conversation with you to learn more about your project, the hopes and wishes, and the impact on ALD patients. So, first, I'd like to ask you, just taking a step back, can you please share your background and what drew you to ALD research? Was there a moment or experience that made this feel urgent to you?
Yeah, well, I started in down a medical path, basically, years ago, with an interest actually in community medicine, family practice, or rural healthcare. I grew up in a rural area, so that was kind of my interest. As I moved along, I became interested in public policy and human rights work. And as I moved into medical school, I became interested in the neurosciences a little bit more and research. Um, it was really a lot of chance encounters that sort of brought most of us into the leukodystrophy field, meaning most of us in the space, with a few notable exceptions, did not have family members affected by the leukodystrophy. So we wound up here for other reasons. And usually it's an encounter with an important mentor of some kind. For me, it was a few people. I'll just name some names here, but there was somebody named Jim Powers, who was a neuropathologist at the University of Rochester, where I was at medical school, who was my first research mentor. He was a really he was a very tough boss, but he was one of the people who described some of the original neuropathology in ALD. Mario van der Knop was considered sort of my research mother. She's a Dutch scientist, and she was really formative in keeping me in the space. I met her through Dr. Bowers, and they connected me with my first research project, and I started working on it from there. And I soon thereafter met Florian Eichler, who became a very important informative mentor, specifically in and around ALD. And then, you start to meet people in the space, and you start to say, hey, I like these people, I like the families, I want to help. And now I'm, a research professor and clinician studying ultra rare diseases at Stanford University as a child neurologist. And so it's a long way from where I imagined I would be as a family practice doctor in rural America. It's like a small town doc. But I think of it in maybe the same ways, in that I don't live in a small town, but I work and live in that same small community, which is the ALD community. And that's my small town. Those are the people I know and I work with and I enjoy interacting with. And so you feel very motivated in your small town to help the people in your community. And so that is still my small town place, just in a different form. And so that is really the thing that keeps me, and all of my colleagues, I really feel the same way. In the rare disease space, you just have this very poignant reminder every time you meet new families or have new encounters with your colleagues that this is a space where children and their parents and their families have really some life and death worries and concerns, and they are scared and hopeful and all of these things, and you want to help. And it's great to feel we all want to feel useful, and being able to feel useful, to feel like the work you do is meaningful and that connects you with people, it's the recipe for a good life. So may we all be so lucky to do something like this, where we make great connections, we do work that we find meaningful, and we also, in the case of doing science, I think science is really fun. So it's like I always describe it as it's a giant puzzle, and so if you like logic riddles and logic problems or puzzles of any kind, it's an infinitely deep place to work and spend your time. It's so for young people who are thinking of coming into the field, that's sort of how I describe it. I say, if you like puzzles, and science is it's one of the best places to work. And if you want to help people, medicine is a beautiful place to spend your career, and you can do both. It's a lot of extra. As a physician scientist, you get to take on a lot more challenges. So you wear more hats, and sometimes it adds more complication to the usual practice of being a doctor. But for most of us, the reason we're here is because we really find it enriching and rewarding and inspiring. It's it's really, I never really imagined I'd be doing really most of anything that I'm currently doing. And but I really feel so lucky. I can't believe I'm doing this job. I get to do this kind of work all the time and work with awesome people, and meet all these really fun kids and their families, and it's just a lot of fun. It's not always fun. I mean, there's some really hard moments too when people get really sick and you can't help, but that's also part of the inspiration to keep moving forward.
Yeah. Well, we are so very grateful to have you in the ALD community, and you are very far from rural Michigan, right? Is that Michigan? That's where
Yeah,
you're
right.
from.
That's where I grew up. Yeah,
I'm living in California now, but I since we joined this community because of our diagnosis, I feel like it's definitely once you get to know people, you start getting to know their LD friend, and it just keeps on going and going. And if all of a sudden I feel like I just know almost everyone by their first name and I know exactly how they got brought into the community. So let's talk a little bit more about the research project. You said science is fun. Yes, science can be fun, but not all of us know that much about science. So in plain language, can you please tell us more about this project? What is it about? What questions are you trying to answer? And what's the biggest obstacle that your work is trying to overcome?
yeah. So the project that we're working on with Cure ALD is a prevention project in a mouse model of ALD that's looking at the ability of one dietary, it's a natural dietary fatty acid called nervonic acid, looking at whether supplementing more of that can prevent or reduce the onset of adrenal problems. And, we're interested in this in terms of adrenal problems and cerebral problems. And there's many avenues to pursue this, but this project is really focused on adrenal issues. And the premise for all of the prevention work is we now have the ability to diagnose ALD at birth when kids are really incredibly healthy and remain healthy for years and years and years. And yet we have no way to kind of act on that in a more proactive way by saying, you have this diagnosis. Now what can we do? And the answer is at the moment, well, it's mostly kind of watch and wait, and we'll tell you if something goes wrong, and then we'll try to treat that when it goes wrong.
Which is so
I think
hard
this is,
for parents and patients.
yeah,
So hard.
it's a bad setup. It is a setup for a lot of anticipated grief. And so it's a worry about something that may or may not ever happen. Our goal here is to at least give people a sense of here's the thing I can do that is reasonable and safe and might help. And that feeling alone is sort of medicine for the family. We hope that the thing for the child is indeed medicine for the child, and that's the nervonic acid. But they actually, having something you can do, there is nothing to combat worry like something proactive. And if you have something that you can do that may help, that's a medicine of its own kind against worry and grief and angst and all of these things that haunt us. That's really what we're looking for here. In this particular study we're going to be doing, we're going to be looking at this medicine or this dietary supplement to see if adding it to the diet of these ALD mice they're just missing the ABCD1 gene, and so they have higher fatty acids. They're not as high as they are in humans, but they're a little bit higher than they are in normal mice, and to see if it can reduce the signs of adrenal injury. And so we plan to do that by measuring some adrenal stress levels that all families will know, ACTH levels, and then also looking at adrenal gland histology and pathology and comparing them between the treated mice and the untreated mice. And important for me to say that this is work that was all pioneered by Dr. Rena Kartha, who's at the University of Minnesota. We're really just partnering with her on this. She's been working on this for a long time, and I've been a big fan of her work and what she's doing. So this is kind of we have a few projects looking at this, and this is one. So I do think that the goal here is to actually help get this possible therapy to a clinical trial. So we hope to use this information to understand, okay, could we even think about using this medicine to prevent adrenals, or this dietary supplement to prevent adrenal insufficiency in a human? And these mouse models help give us just a clear sense of that. They do not predict everything with complete accuracy, but they give us some clues. And that's how we're going to try and use it here. I do think if we ever use this therapy in a clinical trial in ALD, we would probably use it first and foremost to try and see if it can prevent adrenal insufficiency. And typically in prevention models, it's not 100% meaning our goal is to reduce risk. It may not be 100% elimination of all the risk, but even some amount would be a big step forward if we could even reduce the risk by some meaningful amount. If you have a 50% chance of getting adrenal insufficiency by age 10 or age 20, to reduce it to 25% means that a lot of people don't get, would avoid that risk. And yeah, that is really what we're trying to assess here. We're trying to see if this is going to be suitable for a clinical trial. That's really our goal.
That's great. And you already touched on this a little bit, but just so that we're really clear here is for this study, what would success look like? And if this study does go the way that you hope, what changes would you like to see for how it is treated or stopped?
Well, in the short term, success would look like normalization of, adrenal stress levels and adrenal histology in the mouse model. Over the medium term, it'd be, success would be just getting this to a real clinical trial for ALD families, where we could enroll families to have access to Nervonic acid in a safe and controlled way where we could figure out if it really is helping.
That's great. So many families would benefit from this. Just, I know that so many families are excited about this too, because you talk a lot about, it is excruciating to just wait and see. So having something that could be a preventative option would be amazing for families. So speaking of the families, I think it'd be great to hear from you. If you could sit across from a parent who just got their child's diagnosis, what would you want them to know about where this research is headed and how it might help them and impact their family?
Yeah, I would probably lay out, the current paradigm is we get a, we have a diagnosis, and then it's a watch and wait. And what we hope here is that this will give us an option to not just wait passively, but to act proactively, and to see if in the near term, see if the dietary therapy can help reduce the risk of disease. So I think in the near term, the goal is to try and just create access to a new clinical trial or a clinical trial for a new therapy. Whether or not the therapy works it is hard to predict. We think we can model things pretty well. We certainly do our best. In reality, things sometimes get more complicated when you put them in trials. You find maybe it's not quite as effective as you would hope, or maybe there are side effects you weren't expecting, or maybe it doesn't, the effect wears off in some way. And we have thoughts on how all those things might be relevant to this particular, dietary supplement, Nervonic acid. But in reality, it's always a hope but verify kind of thing. We hope it's going to do the things we expect it to do, but it's our job in the scientific and clinical community to try and verify that it's doing the things we hope it will.
Yeah. So, what has brought you to Cure ALD as a funding partner, and what made you want to pursue this work with our support?
Yes, obviously it was meeting Terry and Adam and Theodore and and learning about your family story and providing a project and being very graciously selected for community funding. So it's for the Kickstarter campaign, I guess. So this is really kind of our first time doing that. I think it's the first time we've done anything that's quite this specific, kind of a Kickstarter model where we say this is what we want to do, help us fund it. I think it's a neat experiment. The whole funding model itself is kind of an experiment and a fun thing to do.
It is. It's a great way to engage the community too, right? Like I think I remember talking to you about this and saying like what would be really compelling and get people really excited about taking action and donating to a project that could really, make an impact on our lives. So yeah, we're pretty excited about it. Now, the next question kind of related to that is, for those who do want to see this research project move forward, how can they support you, this research study, and what does that support make possible?
Yeah, well, moral support of any kind is always, not every family can contribute a lot, but financially, I mean, the way you're set up is for financial contributions, and those will go through Cure ALD. They go to the nonprofit foundation at Lucile Packard Hospital, which is associated with Stanford, where I work, and then those funds go to our lab to help fund this project. The project itself, at the moment, is funded exclusively by in this model. So the way we've conceived of this is this will help get the most basic readouts up and running. And the most basic readouts are, can we test it? Can we make the medicine? Can we test it in the mouse? Can we measure these blood levels and the adrenal gland levels, and how does that look on a more the more funding we get, the more detailed the analysis we can do. One of the things that's proven pretty interesting and useful for us is our more complex. I mean, to try to avoid using too much scientific lingo here, but to avoid, let's see, what these are called spatial. They basically provide kind of a picture of the tissue. In this case, it'd be the adrenal gland, and then we can kind of measure all of the genes that are active within each cell on that tissue. That's kind of one of the state of the art right now. It's called kind of spatial omics, spatial transcriptomics in this case, and it tells us all the genes that are turned on or turned off, and that has proven really helpful in some of our other studies of the brain and understanding what's happening before and after different treatments or exposure. And that's what we would like to do here. That's not kind of included in our current budget of 30. I think our target is $30,000. Those studies cost actually about that much in and of themselves. Are quite expensive when you bunch them together. But versions of that we can do at different costs and kind of work up to it. And I think the more funding we get, kind of the further along that road we get, or the more, one of the things you need to do to make sure what you see is not just kind of for lack of a better term, just luck.
Yeah.
You'd be unlucky, or you may be lucky in finding that it is really there, or really not, or maybe not there. You need to do it more than once. You need to repeat it. And in the world of science, it's replication. You want to replicate the results. If you can replicate the results, that gives us more confidence that what we see is real. And we really do see things sometimes that look very promising at the beginning, and we repeat them and you don't get the same results. And I'm sure we miss things in just the same way. We run studies that are too small. This certainly happens in clinical trials, where you run a study that's too small to detect the thing that matters to you. And there are statistical things we do to try and make sure our studies are big enough, that we run enough times, and so on. But I remember when I was in medical school, I was surprised, even a little bit shocked, when I learned that there was that tests were not always, this sounds very simple, but that tests were not just a yes or no answer, and that trials were not always a yes or no answer. And there was a lot of ambiguity sometimes in how things get read out. The reality is that biology is, and science in general, can be very messy, because there are lots of things that can affect what we think we're looking at. Even though we think we understand everything we're looking at, there are things that we are blind to at any one time, that we are not aware of or cannot see. And those can affect the way things look scientifically. So as we move forward on these sorts of experiments, we try to make sure that we're not fooling ourselves, first and foremost, and that we make no illusions about how we've learned things and how certain we are, and so on. This is one of the things that makes scientific communication really hard, is that most of the time, to be literal, we're almost never 100% certain, meaning it's always a 98% or 99%, and then it's just an increasing level of certainty. But we have to sort of simplify things, or it gets sort of paralyzing. Sometimes we say, yes, we're sure about this, even though there's always some small chance that the thing we're sure about is, in fact, it could be affected by something that we cannot predict.
That's so hard. I just want black and white answers.
Yeah. Well,
Especially as a
that's
parent, right? You're like, I don't really want cloudy with a chance of meatballs.
Yeah, sometimes it just makes sense to simplify it to black and white. And I think sometimes just for our own sanity, we have to do that, And so that I do think that's necessary sometimes. It is also one of the things that makes, I think the thing that matters so much is just transparency too, is you want to be, transparent in what you're saying. You also have to keep it a level where you don't lose people's interest. If we explain every decimal point, path down that long to the 100th place or something in the decimal points, people lose interest.
Yeah, that's when science doesn't become so fun.
Yeah. Well, it's helpful to me, and my wife's eyes glaze over very quickly when I'm talking. She's not in science, and so it's helpful to have a feedback loop that keeps me speaking in simple terms, or simpler terms that more people are going to relate to.
That's what it's like when I have to listen to Adam talk about his work in finance and bonds. I think he just loses me after like 5 seconds.
Yeah, right. Right. Well, it's helpful for keeps all of us kind of
Yeah.
honest. But it's that way, we're all that way to different things. And
Yeah.
for those of us who are working in one place, we're interested and usually, hopefully, excited about what we do.
Yeah.
Keith, what happens if our funding for this research falls short? Where does that leave us in our journey to finding a cure for ALD?
Yeah, if our funding were to fall short for this particular study, we should be able to accomplish some of the very basics of just getting the study up and running, getting some basic readouts, and we can save some of the analyses for some future funding. So we could save the tissues, the blood, and the adrenal glands for a future analysis. So many of the samples are simply, we can put it in the freezer literally until later and take it out when funding is available.
Awesome. That's great to hear.
Getting the basics up shouldn't be too expensive. We should be able to do that, I think, with some of the basics of the funding.
Awesome. For you personally, what would it mean for this project to complete its research and really just push the envelope here with Curial?
Yeah, each step of the project would have kind of its own level of excitement and reward. And I think step one is getting things funded. Step two is running the experiments. You hope that the experiments run without any glitches. You get the analysis back. You really just want a true analysis. You want to know for real if things worked or did not work. And assuming things work as planned, that's exciting. We can do additional analyses to maybe understand how things worked, which is not always or typically not what these trials are necessarily designed for. You just want to know, kind of like if you were just saying, well, give me the bottom line. Did it work? And that's step one. Understanding how it worked really does matter for understanding either if things went really well or things went really wrong, how you can improve on something in the future, to improve a therapy, to improve a design. And then I think we would really be most excited is if this really helped us get to a clinical trial for Nervonic acid for boys and families affected. I mean, that really is, I think, would be very exciting to me, to Dr. Kartha, I'm sure all the people who are supporting the effort and the families affected, just to have a trial available that matters a lot.
Thank you, Keith. Is there anything else you want to share about this project? I mean, we had our list of questions, but if there's anything that you want to share that other families might want to hear or the community might want to hear?
I would just say thank you. I mean, if this is a neat it's a neat experiment that, Cure ALD is running and trying that Kickstarter-like model of funding where you see what people are excited about and let people get involved in directly in the research process to some extent. I think it's actually something we've not tried in our community before. I think it's a kind of a neat thing. We're going to be able to do it here. So thank you, Terry, and to you and your family for getting things going and trying this out. This was a new idea that I'm just excited to see it come to fruition, and I really appreciate the community support.